Welcome to the differential gene expression module, where we're going to learn about the regulation of transcription. In this talk, we'll first learn about the different mechanisms of differential gene expression and concentrate on how transcription is regulated in cells. We will look at the role of DNA methylation as well as other mechanisms like regulatory DNA sequences as well as histone modifications. Let us look at the different mechanisms of differential gene expression. Gene expression can be controlled at different levels, and the first level is at the transcriptional level, where we can regulate RNA synthesis and thereby control protein production in a cell. Another way to control gene expression is by controlling RNA processing. We all know that the nuclear RNA made in eukaryotic cells has to be processed to form messenger RNA, and thus by regulating RNA processing, we can regulate the protein production in a cell. Another mechanism of regulating gene expression is to regulate the translocation of the messenger RNA from the nucleus to the cytoplasm. Translation is another step of regulating gene expression because it is the step where we can regulate protein synthesis from the messenger RNA that has been made. Finally, just by making a protein does not ensure that it will actually be functional. In some proteins, post-translational modifications are required, and thus we can regulate protein function by the addition or removal of certain moieties like phosphate groups or other types of functional groups. We're now going to look at how we can regulate differential gene expression based on the type of promoters that are present on genes. So we're now looking at regulation of gene expression at the transcriptional level. Now there are two main types of promoters that have been discovered. The first one that we're going to look at are the high CpG content promoters, or HCPs. They are known to regulate the synthesis of transcription factors and developmental regulatory proteins. In the case of the high CpG content promoters, the histones are usually modified to allow transcription to happen. Thus, in the default state, these genes are usually transcribed, so the promoters are normally accessible to the transcriptional machinery, and hence the genes can be transcribed. You could have certain types of histone modification that can start pausing the process of RNA polymerase II, and there are situations where the histone modifications can result in repression of gene expression because the promoter is no longer accessible. Thus, the point to remember about the high CpG content promoters is that their default state is the active state, and hence they are normally accessible promoters. However, they can be inactivated through histone modifications. The second type of promoters are the low CpG content promoters. These are usually found in genes whose products characterize mature cells. In the case of low CpG content promoters, the DNA is normally methylated, and hence transcription is repressed. If we look at the figure, in the case of the low CpG content promoters, because of methylation of the DNA, transcription normally does not happen, and hence the gene is in a repressed state. This is usually the default state of the genes that are associated with the low CpG content promoters. However, if the methyl group is removed from the DNA, this demethylation results in modification of the histones. And these modifications then ultimately result in the DNA becoming more accessible, and thus we can now have the activation of transcription of genes that are under the control of the low CpG content promoters, because RNA polymerase II is now accessible to the promoter. And thus, in the case of low CpG content promoters, the point to remember is that they are normally not expressed because their DNA is methylated. So now that we've introduced the concept of DNA methylation, let's look at what exactly happens in that. Now we already know about CpG sequences in DNA, and the cytosine that's present in the CpG sequence can be methylated to form 5-methylcytosine as shown in the figure below. The methylation of cytosines in the CpG islands of a gene promoter normally correlates with repression. So to look at an example, let us look at a particular promoter of these genes. Here, as we can see in the upper panel, we are looking at two different genes, the epsilon globin gene and the gamma globin gene. In this particular case, the gamma globin gene that is towards the right of the figure has a promoter that is methylated, and hence it is not expressed and is in the inactive state. However, the epsilon globin gene, when we look at its promoter, which is towards the left, it is not methylated, and hence epsilon globin protein is made. In the case of a 12-week embryo, now we can observe that the epsilon globin gene's promoter is methylated, and hence it is inactive and no transcription occurs. Hence, there is no protein of epsilon globin that is made. On the other hand, the gamma globin gene's promoter is no longer methylated, and hence it is now transcriptionally active. Thus, DNA methylation can modulate transcription of genes. The mechanism by which DNA methylation can cause repression is by preventing the binding of certain transcription factors. So when we look at the figure, we can see that there is a region in the DNA where a certain transcription factor called EGR1 tends to bind, and it allows transcription of the gene. However, if the sequence to which EGR1 binds, if there are methyl groups on the cytosine, the transcription factor is unable to bind, and hence the promoter is not able to recruit RNA polymerase and the gene does not get transcribed. Another mechanism of repression is that there is recruitment of histone methyltransferases or histone deacetylases to the chromatin, and this promotes the repression of transcription. So when we look at the figure that's shown below, we can see that the presence of a methyl group on the DNA recruits the protein MECP2. MECP2 is a methylase. What it also does is, since it binds to the methylated DNA, MECP2 promotes the deacetylation of the histone tails. And that results in also the recruitment of a histone methyltransferase that can now add methyl groups to the histone tails, and thereby transcription is repressed.
Now, DNA methylation is usually associated with the repression of transcription, which leads to the silencing of the gene. However, there is growing evidence that in some cases, DNA methylation can actually activate gene expression. It is usually able to activate gene expression by preventing proteins that act as repressors from binding to DNA. Thus, it should be remembered that while DNA methylation is usually associated with the repression of transcription, there are cases where it can also activate transcription.
What is remarkable is that DNA methylation can be transferred to succeeding generations of cells by a class of enzymes called DNA methyltransferases, or DNMT. In this example, what we're going to see is one of the DNA methyltransferases, which is DNMT3, is the enzyme that can add methyl groups to cytosines that have not been methylated previously. So that is what we are seeing in the figure that is shown in the blue box. Now it so happens that another family member of this enzyme, which is DNMT1, can recognize methylated cytosines on one strand of DNA. And when DNA replication occurs, it can actually place methyl groups on the newly synthesized strand right opposite to it, as shown in the figure in the blue box. Thus, the actions of the DNA methyltransferases can maintain DNA methylation patterns across generations due to the fact that it is adding methyl groups after the replication of DNA. The inheritance of DNA methylation can explain one of the genetic phenomena that is observed, which is genomic imprinting. Genomic imprinting is a phenomenon where there is differential expression of a gene, and this expression of the gene depends on the sex of the parent that provided the gene. This phenomenon can be accounted by the process of DNA methylation, and one of the most well-studied genes is the IGF2 gene. It has been found that the IGF2 gene from a father is expressed in the progeny, but the IGF2 gene obtained from the mother is usually not expressed. To explain this, let's look at the figure. Here we're looking at the DNA sequence that has the IGF2 gene that is obtained from the mother, or it is in the egg-derived chromosome. In this case, what we can see is that there is a DNA sequence called as the DMR, where an insulator protein can bind. And thus the DMR actually functions as an insulator, and hence it prevents the expression of IGF2. However, when we look at the chromosome that is derived from the paternal parent, in this case, what we can see is the sperm-derived chromosome from the father has a methyl group on the DMR region. The presence of these methyl groups that is passed on from cell to cell during cell division, it does not allow the binding of the insulator protein to the DMR sequence. Without the presence of the insulator protein, the IGF2 is no longer repressed, and hence it can be transcribed. Thus, DNA methylation can account for the genomic imprinting phenomenon of the expression of IGF2 gene.
In addition to DNA methylation, transcription can also be regulated by other mechanisms. These include the presence of regulatory DNA elements like enhancers, silencers, or insulators. Additionally, histone modifications can also regulate transcription. The commonly found histone modifications include acetylation, phosphorylation, and methylation. These modifications also play an important role in regulating the transcription of genes. With this, we come to the end of our talk, where we learned about differential gene expression and how DNA methylation aids in regulating transcription. We then looked at regulatory DNA sequences and histone modifications and how they can also regulate transcription in cells.